Hi, my name is Lucy Shu from Hampton Hall Country Day School, and my project is the spectrophotometric determination of the DNA binding constant of a novel and potent sorrelin derivative. So, beginning in ancient Egypt, plant extracts containing the molecule sorrelin were used to treat vitiligo, which is a skin disease in which a person will have patches of skin without pigment cells, so it'll look like pale splotches on their skin. And sorrelin molecules are also currently employed in clinical settings as photosensitizing agents to treat various skin diseases, such as psoriasis and eczema, in which a person would have red, irritated, scaly patches of red skin. And this treatment is known as photochemotherapy or PUVA, and it involves a sorrelin treatment. It could be either topical, so they're applying a cream to their skin, or an oral pill, so they're taking it as a pill, they're ingesting it, and it is introduced into the bloodstream, followed by exposure to UVA radiation, so rays between 320 and 400 nanometers. So sorrelin molecule is the molecule that my presentation revolves around, and they are planar molecules that can intercalate between double-stranded DNA base pairs. If you look at the picture down at the right, you can see the intercalation with the sorrelin molecule in between those base pairs, so it's inserted between them. And when struck with UVA rays, they cross-link the double strands, inhibiting DNA synthesis and cell division. You can see it's actually forming bonds with the DNA base pairs. So it inhibits DNA synthesis by inhibiting the DNA unwinding, which prevents cell replication. And sorrelin molecules have been found to have clinical efficacy in treating various hyperproliferative skin disorders, as mentioned before, as well as some cancers. And these are a couple of known sorrelin derivatives, 8-methoxysorrelin, also known as 8-MOP, which has an additional methoxy group right here. Aminomethyl trimethylsorrelin, also known as AMT, which has an additional aminomethyl and 3 additional methyl groups. And this is one of the most potent sorrelins known today. So the addition of these additional side chains, functional groups, enhances the ability of sorrelin to interact with DNA, as the side groups allow better intercalation with DNA. And both of these molecules are currently used in clinical treatment for skin diseases. In collaboration with our colleagues at the Duke University Small Molecule Synthesis Facility, we have designed a novel sorrelin molecule, 6E, and it has several important features of an ideal reagent for photochemotherapy, which is the technique to treat skin diseases that I mentioned before. The planar sorrelin moiety is nonpolar, allowing it to readily intercalate with DNA bases, which, as we know, are paired based off hydrogen bonds, and side chains with acid groups using the Lewis definition of acids, like NH3+, interact with the negatively charged phosphate backbone of DNA to further stabilize sorrelin DNA interactions. Specifically, 6E contains the acid side chain C3H6NH3+, but it can also be prepared as a salt, which is why you see the Cl in the corner down there. Our colleagues at the Duke University also screened a library of 73 newly synthesized sorrelins for enhanced UV-dependent cytotoxicity, and you can see the results at the right. 6C in particular is around the top. It's the second one at the top, and it was selected for its relatively high cytotoxicity when screened for the ability to induce cell death. And we can see that it has a higher cytotoxicity than sorrelin, 8-MOP, and AMT, which were the 3 sorrelin derivatives that I mentioned before. So my goal with this project was to determine the DNA binding constant of 6E, in particular using 2 methods: fluorescence spectroscopy and equilibrium dialysis with UV absorbance spectroscopy, to experimentally determine its binding constant to target DNA molecules, specifically AT40, because sorrelins are able to best intercalate with adenine and thymine. And this would allow us to see whether the high toxicity correlates with a large DNA binding constant and elucidate the mechanism with which sorrelin induces cell death. And this would also indicate whether the acid side chain increases DNA affinity and enhances sorrelin intercalation. With fluorescence, only sorrelin is fluorescent. DNA is not. So any changes in the fluorescence intensity can be attributed to intercalation of the sorrelin with DNA. As for UV absorbance, the DNA and sorrelin UV spectra overlap. So that is why we use equilibrium dialysis, as it allows us to isolate the sorrelin absorbance to analyze the variation in absorbance upon adding increasing concentrations of DNA. Specifically, going into equilibrium dialysis, if we look at the before The side with the black would be the sorlin molecules, and with the gray would be the DNA molecules. And it has a semi-permeable membrane in between with a molecular weight cutoff of 10K Daltons, and the molecular weight of DNA, AT40, is around double that. So we know that the DNA cannot move in between the membranes, but the sorlin molecule, which has a much lower molecular weight, is able to move through. So after putting the plate with both sides into the plate rotator, which spins the plate to reach equilibrium, it takes around 24 hours, it'll reach the right side, where you see some of the sorlin would intercalate with the DNA, and the free sorlin in the concentration would be free to equilibrate, and you would see that as the sorlin moves to the side of the DNA, there would be a lower concentration on the side with just the free sorlin. The instruments for detection, I used the fluorimeter. You can see that AMT has a blue fluorescence, and the UV spectrometer, the red tide. And this is the equation to determine the DNA binding constant, and this is based on the variation in absorbance/fluorescent at the peak wavelength of the corresponding molecule. And the plots of A0 over A minus A0 versus 1 over DNA will be linear, and the DNA binding constant can be calculated from the ratio of the intercept to the slope. Looking at the experimental control, AMT, which has a known binding constant, you can see that with 0 microliters of DNA, it has 1993 counts, and then as you gradually add more DNA, this peak will get lower and lower and lower until you see it with 20 microliters of DNA at 1206 counts, so there's around like a 700 count difference. And using that data, we are able to plot the line and experimentally determine the Kb of AMT, which was around 143,000, which is similar to the literature value of 150,000. And this confirms that this technique can be used to find the DNA binding constant. So then we proceed with 6E, but 6E was actually found to have a pretty low fluorescence intensity, and the change in fluorescence intensity upon adding DNA was negligible, and therefore couldn't be used to calculate Kb. You can see with 0 microliters of DNA, it's at 818 counts. If you remember with AMT, that was more around like 2,000 counts, so it is significantly lower. And then after adding 20 microliters of DNA, which was the same amount added to AMT, there's 787 counts. That's only like a 20 count difference. So since this change wasn't that significant, we weren't able to find a DNA binding constant. So since fluorescence was unable to produce sufficient data to determine a binding constant, we need to use another method, UV absorbance spectroscopy. And in order to isolate the changes in sorlin spectra, I used equilibrium dialysis. As I mentioned before, only the side with the free sorlin is analyzed, so there wouldn't be any interference from the absorbance spectrum of DNA. So this is the data for the experimental control AMT. We can see the experimental value is 104,000 and the literature value is 150,000. Looking at our curves, you can see as you add DNA, the peak goes down. And there's a bit of a difference between the experimental Kb and the literature value, but they're in the same relative magnitude, so we can use that as a basis to compare 6E with. I also used another experimental control 8-MOP, which also has a known DNA binding constant, and this had an experimental Kb of 141 compared with a literature value of 700. Once again, in the relative magnitudes, we can use it as a basis of comparison to compare the relative DNA binding constant of 6E with these two controls. And then with our novel sorlin compound 6E, I did 2 trials, which you can see here, and then using the average of those data points, I was able to get the line and find the experimental Kb of around 39,300. And looking at a table of them all together, comparing experimental Kb with literature values, we have our 2 experimental controls and our molecule 6E. I also included unmodified sorlin in this table, even though I didn't find an experimental Kb, because it's a good reference to compare unmodified sorlin with sorlin derivatives that have additional groups. So we can see that 6E's experimental Kb lies in between the experimental Kb of AMT and 8-MOP, and this suggests that the additional aminopropoxy side chain of 6E did in fact increase the DNA affinity compared to 8-MOP. And we can see that the experimental Kb is also larger than that of the literature value of unmodified sorlin. So that means that the side chain does increase the DNA affinity compared to unmodified sorlin, and it was more effective than that methoxy group of 8-MOP in enhancing sorlin intercalation because it has a significantly higher Kb. So the experimental Kb of 6E was greater than that of unmodified sorlin and 8-MOP, both of which we saw at the beginning had relatively low cytotoxicities. So this indicates that a high toxicity generally does correlate with a large DNA binding constant. So that suggests that, oh, thank you, that intercalation of sorlin with DNA does have a role in high cytotoxicity. And we see that the acid side chain that we added to 6E does increase DNA affinity and enhances sorlin intercalation. Though 6E was shown to have a higher cytotoxicity than AMT, we just saw that it had a lower DNA binding constant than AMT, and this suggests that other factors, aside from DNA affinity, may be relevant in sorlin's ability to induce cell death. So it could be the photoadducts and other cell components, such as the proteins or the membrane, and it would form photoadducts with those cell components upon UVA irradiation that may contribute to its ability to induce cell death. Ultimately, adding acid side chains may help optimize the sorlin molecule to take advantage of its full therapeutic potential, and this could prove to have therapeutic relevance in improving skin disease treatment, as 8-MOP, sorlin, and AMT are all used in clinical settings to treat skin diseases. So this shows that 6E would be a great candidate to be used for that as well. And our future work would involve investigating the role of photoadducts with other biological moieties, proteins, and membranes in cytotoxicity to determine exactly what causes sorlins to be so effective in inducing cell death. Thank you.